Okay, it's my pleasure to introduce our speaker today for our session of FAANG prediction. Dr. Oscar González Recio is a professor at Roslin Institute. He's currently the chair of the microbiome section of ASAG, International Society of Animal Genetics, and also a member of WCGAP, World Congress of Genetics Applied to Animal Production. And he's also a research member of the Feed and Gas Working Group of the International Animal Data Control Community. Oscar, please go ahead with your talk. Thanks.
Thank you, Hao. It's a pleasure to be here today with you, and I'm talking about prediction of complex traits. I tried to start sharing my presentation. Can you see my full screen now?
Yeah, it works.
Okay, great. Okay, so the title of my presentation is Challenges in Complex Traits Predictions and the Use of Intermediate Phenotypes. We know that predicting complex traits is central in animal breeding to detect animals with best genetic merit to disseminate genes in next generations or be used as parents of next generations. It's also important to foresee phenotypic performance of animals, both under different environments or different managements, also for crossbreeding and estimating non-additive effects. So integrating multi-omics layers to improve complex traits prediction has been of interest in the last decades. We've been including genomics data like DNA, SNP genotypes, and so on in complex trait prediction, but what's next in the post-genomic era? And here I will cover indistinctly during this presentation two main topics, the state-of-the-art of predictive statistical models and also the integration of multi-omics in predictions. We know that predicting complex traits is challenging because they are affected by a large number of genes. These genes have a small effect on the phenotype, and they explain a small amount of the genetic variance. The complex traits are also influenced by the environment, and there is a large complexity of the biological system for these traits. There has been evolving methodology to cope with these challenges from selection indices in the half of the 20th century, passing through infinitesimal model and then the BLAP model with the mixed model equations. And early this century, we've started to use genomic selection and all the suite of Bayesian regressions, GBLAP, single-step GBLAP, more complex models like semi-parametric models, machine learning, and now the upgrade of machine learning, which is artificial intelligence. There has been an increased interest of predicting complex traits since the adoption of genomic selection in our field. The proposed mathematical models span a wide variety of assumptions, each with its own technical peculiarities, from simple linear regression to highly complex neural networks. Most common models assume a linear additive relationship of each locus on the phenotype, which is actually unrealistic. Simple linear regressions oversimplify biological reality and ignore complexity of biological systems. More complex models, such as semi-parametric models or machine learning, are considered black box models. However, the suite of BLAP models are also black boxes from my point of view. For instance, we are assuming linear independent additive effect of each locus on the phenotype when we include this genetic additive effect. However, Hill and colleagues demonstrated that this infinitesimal additive model can actually capture epistatic variance. So this looks like quite a quite close to a black box to me. So these black models that we use in genomic selection are also some kind of black box models. Meanwhile, well-recognized black boxes approaches, like all these non-parametric and machine learning approaches, let the data speak for themselves, but in the model's own language. So it is easy to become lost in the technical details of all these models, like reproducing kernel Hilbert space, support vector machine, boosting, random forest, artificial neural networks. I'm not going to get into the details of each of these models because they're explained elsewhere. So I will then say, okay, we have used different statistical modeling, different approaches to predict complex traits, and now we have new sources of omic information, which is another strategy that promises to improve predictive accuracy of complex traits. And here we can mention, of course, genomics, which is the one that we have been using in genomic selections. But there is more and more omics sources of information. For instance, we have transcriptomics, epigenomics, proteomics, metabolomics, metagenomics, and now phenomics using sensor data, imaging, and so on. I think that genomics has been the most effective omics source so far. It's cheap to obtain with the SNP chips, and we have increased the predictive accuracy and of complex traits, and also the predictive accuracy or reliability of breeding values. And other sources of omic information have increased predictive accuracy only modestly. So highly precise prediction remains elusive, especially in low heritable traits. Novel statistical methods, including machine learning or artificial intelligence, have only increased predictive accuracy marginally. We know some examples that are like very promising, but this increase has not been consistent in the literature. And I would like to mention here a little bit about predictive ability. We assess predictive ability using different metrics like Pearson correlation, mean square error, regression coefficient, area under the curve, and so on, between the observed phenotypes and the predictive phenotypes in some set-aside tests that we usually call testing or validation set. And we want predictions that are high accuracy and how precise, and this needs to be well understood when we're talking about assessing predictive ability. Usually this is done using cross-validation, and this was first used in genomic selection in the seminal paper by Mewis and et al. We need to implement a careful design in cross-validation, making sure that there is no information leak from testing to the training sets, and this has not been usually the case in the past, but I think that we are now being more careful, and the papers and the studies that I read try to make more careful design for the cross-validation. We can do this also in prospective studies. This is less frequent and require a proper design. In this kind of studies, we have a population and we predict the breeding values or the phenotype for those complex traits in a candidate population, and then we follow them up towards their life and wait until they have enough reliability in their breeding values or they have their own phenotype to do a forward cross-validation, or we can implement the RR method if the data are available and they allow to do this. This is also a good strategy to do. But of course, not all the data allow to do this forward cross-validation, and we need also proper strategies. So how can we integrate big omics data in predictive models? And here we've been using this type of models in which we have here the genomic information, which is affecting the phenotype here. And then we include our omic information that may be some of the omics that I mentioned, transcriptomics, metabolomics, etc. And we assume that they have also some effect on the phenotype. Okay, in this kind of models, we assume that there is some independence between the genomic information and the other omic source of information. In an algebraical form, we can say that we have the phenotype here, which is dependent on some environmental or systematic effects here, the DNA or the genetic effect of the host, given some kind of function, and depending on how we define this function, we may be talking about regressions, Bayesian regressions, BLAB, single step, support vector machines, etc. And then we add another term here, or several terms, each of them with the omic that we want to include, explaining the phenotype of interest, again assuming any model or dependence of the omic with the phenotype, and then we will be talking about different kind of models, plus some residual. This has been very used in our field to predict complex traits using different sources of information. This may be Bayesian linear regression, similarity matrices, machine learning, etc. There is another way. One of the limitations of this framework is that we are assuming independence between the genetic part and the other omic part, and this is not always the case. So we can set our models in a way in which we have the DNA, the genetic information, which is controlling the phenotype, and it is also affecting the other omic information, which in turn is affecting the phenotype. This is called intermediate phenotypes or mediator trait models. Here we have our trait of interest, which is dependent on systematic and environmental effects, also the genotype, plus some other omic sources of information with some effect on the phenotype, plus some residual. And this omic information is also dependent on some other environmental and systematic effects. They are also controlled by the DNA or the additive effect of the animal, or some genetic effect on the animal. And optionally, they may be controlled also by additional omic sources of information themselves, plus some residual. We have used this type of approach in this paper by Sepulveda et al. And here we used our target trait was methane, and we use just the omic information or microbes as another source of information. So basically metagenomics or microbiome information, using this correlated trait as information. And we observed that using both methane and microbes, like two different traits, and microbes being controlled by the DNA of the animal, we could expect a larger reduction in methane emissions across different scenarios, up to 50%. So a 50% larger reduction on methane if we include this source of omic information in addition to the genetic part. So heritable omics are more relevant for selection purposes than explicative ones. I forgot to say, but the microbiome information is heritable. We know that there are some microbial features with high heritability, and this helps a lot on on increasing the genetic gain of the target trait if they are genetically correlated. And this omic information can be phenomics. It is already well known that more phenotypes implies larger GBB reliability, so and this may increase the correlated response for the target trait. This is basic quantitative genetics theory. We can use correlated traits to increase the genetic gain or the prediction of traits that we are interested in selecting. Phenomics is one of the fields that is now enlarging a lot with sensors, imaging, more traits that we can record in for the animals and in the field. So there are now some scientific questions that remain, and is that, could we achieve equivalent genetic response using omics data other than phenotypes? We know that increasing the amount of phenotypes that we can record can achieve larger genetic progress. But if we could obtain a large enough amount of omic information, and if that omic information has larger heritability than the target trait and has a large genetic correlation with the target trait, could we achieve equivalent genetic response using omic data than using other phenotypes? And is it economically affordable in comparison to cheap phenotypes that we could obtain by usual recording schemes or sensors or the new phenomics era? And can we use this in mediator or black boxes models to achieve larger predictive accuracy of the target traits? We also know that there are some omic sources that are redundant with the genomics information. So how does this affect predictive ability and the expected response to selection when we include these omic sources of information? So we have here several scientific questions that I don't think that we have solved yet, and that we'll need to think about in the next years how we're going to deal with this. There is also difficult to obtain such a large number of data from live animals. For instance, if we are dealing with transcriptomics or metabolomics, it's difficult to obtain a large number of records at the right time and the right tissue of animals in the field. So how can they overcome these limitations, and how can they be better than using other omic source of information like the phenomics? And here I would like to interactuate a little bit with the audience. So I will like you to either scan this QR code or just go to slido.com and just put this number here, this code number. And for you to answer 3 questions that I'm going to show next. So I will give you some time to do this. I will monitor this also here. Okay. I'll give you a few more seconds to be able to log on this web page for this questionnaire. And I will start with this question. So classical quantitative genetic models are constrained by simplified assumptions and trait heritability. So do you think is it possible to overcome the predictability limit imposed by heritable and SNP-based heritability in our models? You know that what we can predict using genomic information is basically what is explained by the genomic information, by the DNA. If the heritability is low, then there is a lot of noise on the phenotype. And what do you think? Do you think it is possible to overcome this limit imposed by heritability? Let's see what most people think. So there are some that are optimistic that we can do this, and some others that are not so sure, and some others that think that this is not possible. Okay, thank you for answering this. I will go to the next question. Is the gain in predictive ability from omics sources such as transcriptomic or proteomics constrained by the redundancy with genomic information? So we know that the transcriptomics or proteomics, they share a lot of information with the DNA. So they are actually partially redundant with genomics. So do you think that we can the gain that we can obtain in predictability from other sources of information is relevant, and we can increase the accuracy of predicting complex traits in this case? Okay, here most people are optimistic that we can do this. Okay, most people think it is. Very good. And then last question. Black box models promise to detach from unrealistic biological assumptions, like I said, linear, additive, independent, and so on. And they promise to capture complex modes of gene action. However, limited advantages and high computational cost has been shown so far. So what do you think it may be the reason that there are no we have not been like very a lot of gain or a lot of advantages so far of in using black box models like neural networks or random forests, machine learning, and so on? Actually, most of the studies that I've seen, the predictive ability with compared with GitLab or basic Bayesian regressions, the increase on these models are actually quite small. So here you may be typing a little bit more. I will give you a few more seconds if you need to finish the answer. Okay, yep. So these are a lot of reasons why black box models may not be actually working as well as we or as they promised. Phenotypes are more complex than we can record, limited omic availability, high dimension of predictors, and yeah, that this is important. I will cover this later on. Altered prediction target of phenotype rather than breeding value. Yeah, that's also true. So there are many reasons that are difficult to model with the statistical approaches that we have available right now, and all of these are good reasons why we're not achieving we're not getting a lot of advantages of using black box models and also omic information, other omics information. Thank you all for answering the questions. So between the most promising opportunities of omics data for prediction of complex traits, I think that we can include phenomics using sensors of images as mediators or correlated traits. And you may challenge this, saying that there are also other omic sources that may be even more promising than phenomics. But so far, I think that phenomics, especially when we use sensors or we collect information that is cheap to obtain and we can record it in large-scale phenotyping, they can be even more promising than other sources of omic information. And also we can, there is a lot of interest or a lot of promise from omics that do not introduce redundancy with genotypes. Other omics that are less redundant with the genotypes, because obtaining genotypes nowadays is quite easy. We have sequencing, we have SNP chips, low-pass sequencing. We have this omic source of information that has been very successful on predicting complex traits, and probably we have achieved a plateau right now. And then we need new sources of information that are not redundant with genotypes. The future, or actually I would say present challenges in predicting complex traits, are overcoming the theoretical limit imposed by heritability, as we mentioned before. Big data and models that are not constrained by this predefined parametric space may be useful, if this is even attainable. But as far as I've been able to read in the literature, there is still this limitation imposed by heritability. We also need sustained efforts in obtaining large-scale phenotyping and more parsimonious approaches with reduced parametric rigidity that could enable the field to move beyond the plateau reached in the post-genomic era of predicting complex traits. Of course, as somebody answered the question, deal with high dimensionality of omics data is also a challenge. We need to include or accommodate the models to accommodate this high dimensionality of omics data with a lot of features, each of them. However, any strategy can only be proved right or wrong with more big data, so we need to replicate independent studies, and also we need well-designed cross-validation strategies that I will mention this later on, but we need cross-validation strategies with no leaking information between the testing and the training set. I will now focus on this third part here, how to deal with high dimensionality of omics data, which is one challenge that I think that many of us are trying to overcome. We know that all these omics sources like genomics, transcriptomics, all of them can provide us with a lot of features, each of them, and we can also include different omic sources in the same model to predict complex traits. All of them have a large number of features, regardless of the omic source that we use. There is an interdependent relationship between features, and they make the models to be less parsimonious, with some hyperdimensionality that end up in some hyperdimensionality problems with this large P small n problem. So here I'm going to explain again the two frameworks that I mentioned before. So we can go to framework 1 and perform feature regression, and then we include here our omic source with all the features, and we need to regress them on the phenotype with a large number of M variables. Or we can use similarity matrices, and then we can use mixed models or any Hilbert space that are themselves more parsimonious regularized models. So this is an option, and this is probably the typical approach that we've been using in the field, including these omic sources. But we can also go to this mediating framework that I mentioned before. We have here the phenotype that is regressed on the systematic variables and environmental effects, the genetic effect, and then we're going to use these omic sources with some effect, each of them, and each of these sources are also dependent on some environmental and systematic effects. They are also dependent on the genetic effect, plus optionally dependent also on other features or other omic sources, plus some residual. The limitations are similar as in the framework before, as the previous framework, and these models may require additional restrictions in the models to ensure identifiability. For instance, sometimes we set the residual covariance to 0 to ensure that the models are identifiable. This is the approach that we have been using in metagenomics, for instance, in this paper by Alejandro Saborío Montero. In this case, we use aggregated variables to reduce the dimensionality of the metagenomic data. These aggregated variables were defined as the principal components variables, or PLS, that capture the omic variability. The first two principal components explain between 1/3 and 50% of the microbiome variability, depending on the data set, because later on we have been replicating or using this approach in other data sets with other collaborators in the Netherlands and also in Australia. The principal components are heritable, ranging from 0.10 to 0.30. And they are genetically correlated with production and efficiency traits. These principal components or these aggregated variables can have a biological interpretation, and here what we did was to calculate the loadings in the principal components, and you can see here the top 50 more relevant genera in principal component 1 and principal component 2. You can see that the taxa, the most relevant taxa in principal component 1 with positive values were all eukaryotes, whereas those with the largest negative loading in the principal component 1 were some viruses and also some bacteria. In principal component 2, we also identified the genera that had a more positive and more negative value in these principal components, and they were mainly bacteria, so prokaryota, and also some archaea. We did the same with the top 50 most relevant KEF pathways, and we also calculated their principal components, and this is the information in principal component 1 and principal component 2. This graph was done by Adrián López Catalina, and you can see that again the most relevant KEFs in principal component 1s with a negative value were KEFs that belong to eukaryotes, and the ones with most positive value, in this case, they were prokaryotes or multi-domain. The ones that had a largest loading on principal component 2, they were from multiple domain or prokaryotes, and with negative value, they mainly were prokaryotes. When we analyzed the KEK pathway, we identified some KEKs that were related or associated with the hydrogen metabolism, which is also associated with methane emission and digestion of feed in the rumen. So they are also providing this biological information, and that's why we think that these principal components resulted to be genetically correlated with important traits like milk production, feed efficiency, and also methane emissions. So we can use these aggregated variables as an intermediate phenotype. In this case, we assume that the additive genetic effect is controlling or is affecting the phenotype, but they are also affecting or controlling the microbiome composition, which in turn is affecting the phenotype. We model this in this algebraical form, in which we have the phenotype here that is controlled by some systematic or environmental effect, by some genetic effect, and is also affected by the principal components. We can use 2, 3, 4 principal components plus some residual. And then the principal components is itself a phenotype which is controlled by some environmental effects, also by the additive effect, and optionally by the other principal components, all the other principal components that we are assuming, plus some residual. So this model, this aggregated variable, reduces the dimensionality of the model, and we can also infer causal relationship between the microbiome and the complex traits, as I showed before. The principal components, as I said before, are heritable between 0.10 and 0.30, and they have a large genetic correlation. I'm not presenting these results, but they're under review. But we have published them in mainly with methane. We have published genetic correlations of these principal components and methane, and it is actually quite large. So we can increase the predictability of complex traits and also increase the expected genetic gains of the using this intermediate traits framework. Finally, to finish my presentation, these are the take-home messages that I would like to deliver today. Predictability of complex traits is currently constrained by different factors, as I said, limitations on current statistical models, also the limitation constrained by the heritability, and also because many of the omics sources that we're using are have redundant information with the genomic information that we are all using at this stage. Phenomics shows to be promising as a mediator trait if we can obtain phenotypes or intermediate phenotypes with enough heritability and that are genetically correlated with the target trait, and where the large-scale phenotyping is possible. Dimensionality reduction strategies like PCA, PLS, etc. are a good way to reduce the dimensionality of the omic sources using aggregated variables in mediator models if they are controlled by the genetic effect and they are genetically correlated with the target trait. Finally, just remember, and I would like always to emphasize that we need proper cross-validation strategies to and compare with the benchmark models to establish what is the gain in the gain of predictive accuracy when we include other omics information rather than the traditional genomics information. Thank you all. If you are still here, and I would like to thank the organizer for inviting me to deliver this seminar, and I'm happy to answer any questions if you may have one.
Thank you, Oscar. So for the next talk. So any questions from our audience? If you want to throw that question to the chat, it's also fine. Yeah, we got one question. Go ahead.
Yep. Thanks a lot for your talk. It was very interesting. As you showed in the graph that obviously there's a redundancy between the genomic and the omics information. and I'm a bit interested about what is not redundant, because biologically speaking, there's no phenotype that is directly produced from the genomic DNA. There are always like intermediates and molecular phenotypes in between, RNA, proteins, and so on. And I was wondering if what is not captured by the omics way, in the graph. So what comes directly from the genome is not actually everything else than what is provided as information, like the actual data. I'm not sure it's clear. Yeah, do you see what I mean?
I think so. So you mean that there may be some information that is not redundant, or you think that everything is every omic source of information that we are using at the moment is redundant with genomics?
No, not it. I don't think it's everything is redundant, but my point is that we never have at our disposition when we train the model, for instance, we don't have all the intermediates and all the omics, right? We only have access to some limited amount of data, for instance, transcriptome information or so on, but we never have everything.
Yeah,
So I'm wondering if the direct link from the genome is not actually capturing everything else, and that at some point, if we had enough information, we wouldn't need anymore the genome information.
Yeah, that's a good question, Sylvain. I think you're right. So if we would have all the information, even if it's redundant, maybe we can perfectly model the phenotype with all the information other than the DNA. And we could achieve the same or even larger accuracy than using the DNA. Then we need to think, should we use, because obtaining a large number of omic sources is expensive. Can we use that in breeding programs? So it depends. I know probably you're not all of you are in the field of animal breeding. I don't know if that's the case, or animal genetics. And then you may have other objectives or other situations in which you will prefer to use other omics informations than genomics, and that's perfectly fine. You may be trying to predict complex traits, for instance, in human diseases or in the clinic. You're not that much interested on breeding animals, and then it may be worth to use other omics information for that. It may be more expensive, but you may have better results than using genomic information. So I think that each problem has different approaches, and I think that you as experts in your field, you need to know, or you need to study or investigate what is the best approach and the different sources of information that you need to use for predicting complex traits. And in that case, you're right. We may be later on, like in the future, having different sources of information that they may be even better than DNA, and they have larger accuracy than the one that we are obtaining at the moment. I don't know. That's something
it's not soon.
For the future.
We're far from that point yet.
Yeah, I think so. Yeah.
Thanks a lot.
Yeah, Mara, go ahead.
Hi, Oscar. Thanks for the talk. I was wondering, for the last couple of things that you presented, when you use those few principal components, would it be very similar to run like a multi-trait model where the principal components are simply correlated traits to the trait of interest, rather than using them as, say, an intermediate phenotype?
Yeah, actually you're right. So these intermediate models, this intermediate framework, is based on the structural equation models that you know, and you can actually set the model like in a multi-trait, in a multivariate statistical approach. You just need to take into account this. If there is some recursive effect of the intermediate trait on the phenotype, then you know that you need to take this into account when you calculate the heritability and genetic correlations, depending on what framework set of equations you're using. You know that you need to carefully consider how you're calculating the heritability and the genetic correlations, because you need to take into account this recursive parameter in that case. But the two models, we have demonstrated that the two models are equivalent. The mediator trait, that they are based on structural equation models, and the bivariate or multivariate approach. I think that using the mediator traits or the structural equation models help understanding what is the effect of the intermediate phenotype on the phenotype. It's more informative, but the models, the set of equations are equivalent.
Okay, thanks. Yeah, thanks.
Yeah, but Maro, that also depends on how many PC we have, huh? It's like that if you go to a large number of PC, then you're going to fit a multi-trait model with a huge number of traits. So probably computationally is also hard. But the one Oscar present, that part is kind of like not there, huh? So computationally might be easy. Yeah.
You need to impose some restrictions too in the model if you want to do that, and yeah, it depends at the end of the day what assumptions you're making on the different equations, how many traits you are analyzing at the moment, and things like that. What covariance you are assuming, if you are assuming that there is residual covariance between the traits or not, and things like that. So at the end of the day, they are equivalent, but how do you implement the models? They make the two approaches not to be equivalent, and then you need to interpret them carefully.
Any other questions? Also, I have a question here. So you keep talking about this kind of redundant information. Is redundancy, let's say, between the omic information and the genomic information only let's say a bad thing, while the redundancy between these genomics and omics information improve let's say the robustness of the model? If you think about it, when genomic selection was introduced, people were fitting the genomic information and the pedigree, the numeric regression matrix they have. So, but genomic information and the pedigree, it's kind of, there's also some redundant information, huh? You use these, yeah, you see my point, huh? But yeah. So, but yeah, that's my point. So it's the redundant information. But from your talk, looks like that you are going to say the redundant one. So we try to get rid of the redundant one, huh? But my feeling will be having it there. And that's my question. Will it improve the robustness of the model, and let's say provide additional layer of information, even though it is redundant, a part of it is redundant? How do you see that?
Yeah, that's a good question too. How there's definitely some redundant information, and I think we need to do a better job on canalizing that redundant information. So, okay, in some way, like extract the redundant information from what is not redundant information, and emphasize somehow the non-redundant information in the omics data. And we probably need to go and develop smart strategies to do this. I don't have the answer for this right now, but maybe instead of using transcripts, then maybe we need to use information on how this DNA is making the transcripts, and how much variability can we extract from the different transcripts, things like that. I don't know if that has been done so far. Same with the proteomics and transcriptomics. There is probably a lot of redundant information between transcriptomics and proteomics. I'm not aware of any study that is including the three sources of information and how much they are adding on the other. So yeah, I cannot answer that question. Definitely there is some redundant information, but there is also some additional information, and how to separate them, I think that's a challenge for the future.
Yeah, I agree. I kind of like also think probably when we talk about predicting the breeding value, or let's say association, try to detect a causal gene, or let's say significant signal, probably the redundancy of also play a different role, huh? You see, huh?
Yep.
So yeah. I kind
Yeah,
of like
definitely. And this enters also in this strategy. So what is your objective? Your objective is to explain the biology under the trait, or your objective is to predict the phenotype or the breeding value? And depending on what is your objective, then maybe the strategy is different. And what is good for one of the to deal with one of the framework is not good for another.
Yeah, great. Do we have any other questions here? Yeah. On the other side, I do have a really a kind of like a practical question. So yeah, given your experience, I've been working with different omic data. Huh? So let's say I'm in industry, I'm in a company. Okay, now I have some budget. Okay, I'm trying to see, okay, with my maybe $500,000 budget or whatever. So I want to see the potential of omic data, but maybe I could only pick one or two, like transcriptomics, a microbiome, epigenome, all this stuff, based on your understanding of, let's say, the redundant information from different omic data, let's say the cost, what's going to be your recommendation? Let's say I have this kind of budget, I can design experiment to see the potential of these data. I have no idea about, but I want to see your suggestion based on your experience. Yeah.
Yeah, well, that's a difficult question. How? I don't know, because it will depend on how many animals, what is your objective. If you're trying to predict complex traits, you're going to implement this in the industry, or you just want to understand the biology undergoing the complex trait. So I cannot provide a simple answer on this. I would think that a transcriptomic is quite redundant on genomics and also proteomics. Metabolomics and or microbiome is less redundant with genomics. Probably I will emphasize on that, or I will rather put my money on those on those less redundant omic information. And also phenomics. I think that phenomics is a world to discover, and is going to be very popular in next year. All this data that we can, or traits that we can obtain at a very low cost using sensors, MIR, spectra, imaging, things that we can implement in the industry at low cost, and that can provide a lot of data that could be a good value for money also for the future, I think.
Great, thank
But
you.
again, if you are interested on the biology undergoing the trait, and how the DNA is transcribed in proteins and things like that, then of course you need transcriptomics and proteomics to do that. So as I said, it depends on your interest.
Mm-hmm. Yeah. Thank you for the answer.
I have one question. Sorry, I'm driving and I can't log in. But interested in animals. Are you guys looking at environmics? Sort of getting more data from sensors on the environment. I guess maybe less of an interest in cattle that are long-lived, but maybe for poultry, if you can predict the environment for the main production season, and get a better prediction of how that genotype performs in a given environment at a certain time. Is that something of interest? I think that's where plants are moving towards.
Yeah, definitely, Sean. That's a very good question. I think there is a lot of interest on this environmics science, not only in poultry but also in plants, in aquaculture as well, where the environment may determine the complex trait, the expression, and many different things in the phenotype. And I think there is also a lot to explore there. Sensors, things like that, imaging as well. As I said, they are technologies that are very are getting a lot of attention on those fields.
Thank you.
And then we will need to think how to model those information in our statistical models. Maybe we need to develop new models or strategy. Artificial intelligence is very promising in the environmentomics science as well. So I think
Yeah, I
that
think
we will
it's.
have. Yep.
Sorry, I think it's similar, right? It's just dimension reduction, trying to identify similarities between environments and identify where genotypes have either peak performance or stable performance. And I think some of these AI techniques that are able to deal with very large sets of data are promising for sure.
Yep. Yeah, definitely. And maybe we need to go back to the two stages approaches that we use at the beginning of selecting features that are of interest or associated to the phenotype. And then there are like a previous step where we select the features that are affecting the phenotypes. And then we model those phenotypes to reduce the dimensionality. Yep, definitely.
Correct. I think one of the big tricks, though, after that as well, is thinking about cross-validation, right? Some of the techniques that you mentioned about on the genotype become very tricky on the environment as well.
Yep. Yeah, definitely. Yeah, that's why I think that cross-validation strategies are very important. Also, because if you have a data set and you're working with some environmental conditions, if you want to predict in different environment, you also need those conditions, and you need to test the models in both conditions. So you need to have information of the environment in both data sets. But at the same time, you need to not leak information from the testing to the training. So the model is only trained on the training set without training hyperparameters or things like that in the whole data set. Otherwise, you may be overestimating the predictive ability.
Correct. Yeah, and I think this becomes a very, very important issue for companies, right, that are trying to fast track their, say, pipeline and release varieties faster, saying that they have this degree of confidence. If you make the wrong decision based on an overestimate of your predictability, you can make a very, very bad decision that will be bad for a company, right? So yeah, it's very important.
Yep.
Great. I think we run out of time. 1 hour. So Oscar, thank you so much. Thank you for the talk, and thank for the audience for the great questions.
Thank you for having me and for the good questions, the good discussion.
Okay, so then